Now, without further ado, I would like to introduce our first speaker, Nevan Krogan. So Nevan is professor of cellular and molecular pharmacology and the director of QBI. Thank you, Nevan.
All right, thank you, David. So I'm going to give a brief overview of an initiative that has been started from the Quantitative Biosciences Institute called QCRG, QBI Coronavirus Research Group. Next slide, please. So a group of 22 different investigators at QBI have come together over the last month, I would argue in an unprecedented collaborative way. This includes systems biologists, structural biologists, biochemists, chemical biologists, computer scientists, virologists, and we're all working together, bringing our diverse set of skills together to try to help in this particular pandemic. Next slide. And one of the things that we're looking at is trying to target host factors as a potential therapeutic strategy. There's a lot of work ongoing, obviously, where people are trying to target the viral proteins, which is great. We're taking a different tact. And one of the things that we're doing is, we've done is generated a SARS-2 human protein-protein interaction map using affinity tag purification mass spec-based approach. This map is fueling hypothesis-driven research using biochemistry and structural tools led by Dave Agard and Clem Verba, Oren Rosenberg, Bob Stroud, Charlie Craig. Also, the chemical biologists are looking into this map, as you'll hear about, including Jack Taunton, Matt Jacobson, Nevan Krogan, Brian Shoykat, and then obviously bioinformatics. And the fact that we got to work remotely now, this has been great. And we've been collaborating also in this regard with scientists in Seattle, San Diego, and England. And the goal here is to put all this information together to predict key host factors that are involved in viral replication, perturb those pharmacologically or genetically, and then look in infection experiments to see what effects these perturbations have on infection. And then in a reiterative way, this reinforms these other experiments that are going on. Next slide. So when the sequence was released January the 24th, Dave Gordon and my group led an effort where he cloned out, or tried to clone out, each one of the 29 predicted genes. And we put affinity tags on them. The Western blots are on the bottom. And what we've done here over the last several weeks, this is a project that usually takes a couple years since we've all got together to work collaboratively. It's taken a couple weeks where we've expressed each one of these proteins in human cells and HEC293 cells, purified them, and then analyzed the co-purifying material by mass spectrometry to identify the host proteins that are physically associated with each one of the viral proteins. And I just want to say, since we released this study a couple days ago in bioRxiv, there's been over 50 laboratories that have requested these constructs, and we're sending them out. I think I got another 10 emails this morning. So we're sending out these reagents to scientists all over the world. Next slide. So during the course of this study, if you look on the left-hand side, we identified 332 different human proteins physically associated with 26 of different SARS-2 proteins. And we've been doing a number of different computational analyses with this. I just wanted to show you one briefly. If you look at the 332 human proteins and you look at expression across all different human cell types, it was actually very exciting to see that these 332 proteins are most highly expressed in lung cells. And even though we're looking in kidney cells, HEC293, the fact that we're enriching for proteins that are highly expressed in lung cells give us confidence that we're identifying physiologically relevant connections. Next slide. So here's the map that we have generated and released to the scientific community as of a couple days ago. We encourage people to look at this map. If you want to reach out to us to get this information, we're happy to share with you. Next slide. And scientists in the QCRG are already delving deep into these connections, making hypotheses on them. Obviously, I'm not going to go into all of this information here, but using modeling approaches, structural-based approaches, we've started to look deeply at some of these interesting connections. And this is work being done by David De Ruggiero, Stephen Flor, John Gross, Tanya Cortemi, Andre Shalley, James Fraser, and others. And now what we're doing is ordering reagents so that we can test these predictions experimentally when scientists can more freely go back into the laboratory. Next slide, please. And efforts out of Nevan Krogan and Brian Shoykat's lab, they've looked at these 332 proteins and have now made predictions about FDA-approved drugs or compounds that are in clinical or preclinical studies. And they've identified 69 different drugs and compounds that could potentially target the human proteins that we find physically associated with the viral factors. And in this set of 69, there's 27 FDA-approved drugs. Interestingly, one of them is actually chloroquine itself. Brian predicted that it actually targets a human protein. And now what we need to do is test these in virological assays. And one thing I just wanted to say, when we released this, we filed no IP on any of the interactions or any of these compounds so that the information is freely available to accelerate the discovery of a treatment. Next slide. So unfortunately, we don't have the virus propagating at UCSF. I know Melanie Ott is working very hard, as you heard, at the Gladstone Institute to get this within the next month. Fortunately, we have some really great collaborators around the world who do have the virus propagating, including Adolfo at Mount Sinai in New York, and then Marco and Olivier at the Pasteur Institute in Paris. Actually, Marco actually got infected with this virus a couple of weeks ago, but he's now back in the lab working. And Kayvon and Brian sent out now 22 of these compounds, and they're presently being tested. In the next few days, we're going to have results in a cell system to see what effects they have on infection. Adolfo has a ferret model now working, so then we can extend these type of analyses to a mammalian model very quickly. And the rest of these compounds are going to be sent to them in the very near future. Next slide. This is a paper that was put together, I'd argue, in an unprecedented speed as a testament to the collaborative effort. This was released on bioRxiv Sunday night or Monday morning, I don't even remember. And I'd encourage people to look at this information. And if there are any more details or reagents that you need based on the study, please let us know. Next slide. And we've been coordinating our discovery research efforts via Zoom, just like this, every couple of days. Sometimes we have over 100 basic scientists or discovery scientists on these Zoom calls. And this has actually been quite exciting to me of how much we can get done remotely. Next slide. And we've assembled these different subgroups so that we can be more efficient. So for example, Klim Verba, Oren Rosenberg are running the structural biology subgroup of QCGR, Cherie, the protein trafficking group, Denitsa Fujimori and Alan Ashworth, the epigenetics group, and Natalia Jura is focused on the mitochondrial connections that have come out of this particular map. So we've been meeting regularly, and one of our goals in the discovery research community in a week or two is to have a town hall for everybody so they can see what we've been doing and how we can connect to other entities at UCSF. Next slide. And the last thing I'll say is, we've been trying to get together on the discovery research end. We're very excited now to be connecting more to the clinical world and the immunologists as well. And hopefully the call today will help ultimately facilitate that. And the last thing I just wanted to say, it's been such a heroic effort by so many scientists. I just want to name a few people in my group: Dave and Gwen and Jeff and Mehdi and Rob and Ruth and Danielle, and so many other scientists in all these different labs. It's truly been a heroic effort, and it's been awe-inspiring to me to see how people have come together in such an unprecedented way. And finally, I'd just like to thank Kayvon Shokat and Brian Shoykit. We've been working around the clock on this for the last month. Thank you.